Ooh, that sounds loud.
Do we need to adjust volume for you, or are you getting feedback?
I'm getting pretty remarkable feedback. And I'm using headphones even.
So your voice is not that loud in the room here. So there may be...
Is the sound coming straight out of the computer or something?
All right, I adjusted it down a little bit. Let's see if that makes a difference.
All right. It's better.
Yeah, go ahead. We'll adjust it on the fly if we have to.
Okay, that sounds fine. Excellent. Well, it's a pleasure to be presenting on this bizarre screen sharing YouTube feed and everything else going on. So the story that I'm going to talk about today is less about the molecular aspects that I talked about in the past and more about the phenomena that we've got, I think, a better handle on than we ever had before. Can you hear me okay?
We can hear you fine.
Oh man, the echo is pretty fantastic on my end.
That must be your microphone then. You keep talking and I'll play with your mic.
All right, sounds good. So this is definitely work with a number of people, but notably Jessica Lee and Siavash Riazi, especially Jessica. And I think she's really responsible for some of the key insights that we'll be talking about today. And so mainly it's been the two of them, but then also at the end I'll talk about the work that Janelle and Tomislav are doing that will be in collaboration with both Jeff's and Ed's lab in terms of looking at transcriptomics and proteomic work. All right, so just as a very quick review, or for people who might be new, a new picture of this, what I'll be talking about today is formaldehyde as a toxin. And the bug that I study, the phylobacterium, can eat methanol as a sole source of carbonated energy. And as you can see in this overly complicated picture of metabolic pathways, formaldehyde is made from methanol in the very first reaction, and then gets further converted through a series of steps to formate, and that serves as the major branch point between CO2 and going into biomass. And so even though when they grow on methanol, which they do very quickly, they produce formaldehyde at about 2 millimolar a second. If you give them formaldehyde from the outside on top of methanol media, even numbers above 5 millimolar suddenly start causing pretty rapid death. And so you can see the chart over on the side where there's the 5 millimolar going through to 20 millimolar and seeing rather rapid exponential decline in death over just a series of a couple hours. Actually, a lot faster than a lot of antibiotics kill. And so it's been a big question in terms of how they can handle having such a nasty toxin. It's something that they convert all of their food into. And so that was looking at formaldehyde kind of 5 going up to 20. If you look at smaller levels of formaldehyde in the media, if you add either no formaldehyde, 1 millimolar, 2 millimolar, growth on methanol is unaffected. You get to like 3 or 4 millimolar. What you see is by the time you observe growth looks fairly typical, but there's this very long lag. And at 5, only some fat ever recover. And beyond that, we never see anything.
That's really just a twofold difference, inducing that dramatic lag?
Yes, yeah. And you can see by the time they start growing, everything looks totally normal. But there's just this really, many hours or even days worth of lag. And so we thought that the lag was a lag, but it's not. During this period of time, there's actually a substantial amount of death and then a substantial amount of regrowth. And so you can see at 4 millimolar, for example, that they die by about 3 orders of magnitude over the first 15 or so hours, and then they regrow at an exponential rate that's actually basically the same as their normal growth rate. And so the critical question that arose was, why did the cells change from rapid growth, and I should have said rapid death, from rapid death to typical growth rates? And so there's a few possible hypotheses, and I think it's worth kind of enumerating what these are, then we'll talk about kind of how we ruled out the various ones. And so one possibility is that they're actually different cells. that during this period of time, we've selected for genetic resistance. We know, for example, and everything else I've ever told to you guys, about EFGA, this formaldehyde regulator, that if you knock it out, they can grow in the presence of formaldehyde. So it's possible that this is just the easy occurrence of loss of functional alleles in EFGA. It's also possible that over this period of time, they initially can eat the formaldehyde down until it gets down to a threshold where they can just barely take it and they start to grow again. So the reason why you go from death to growth is because the world has changed. It's also possible that since we're looking at viable cells, that maybe not being able to make a CFU is not actually the only way of thinking about life or dead. So that those cells that aren't making CFU are only partly dead, and then they recover. And in many areas of environmental microbiology, this has a name. This is viable but not culturable. And so it's possible that they're partly dying and then partly undying. And that represents these two phases of this curve. And then lastly, there could be a phenotypic subpopulation that is more resistant than the others and survives, and actually not just survives, but actually grows despite lethal levels of formaldehyde. So those are kind of, I think, the full topology of possible ways to see this kind of death and then regrowth. And so, in terms of number one, that it's a genetic resistance, that we're applying a stress, only the rare survive, and then once you remove, those are the ones, this is not it. You can take cells, we've done this with many individual colonies that have regrown, grow them up again, test this experiment, and you get the identical phenomena. They have not become formaldehyde resistant. And so there are no genetic mutations that seem to explain what's going on. So the formaldehyde threshold model was very appealing. We thought perhaps if you follow kind of formaldehyde concentration as a function of time, perhaps as the cells are dying, they're also chewing down the formaldehyde, and it gets below some threshold level between kind of net death and net growth, which is when you see this kind of change, this inflection, and then they start to grow again because the formaldehyde's low enough that you can take it. And so as long as they can get the formaldehyde below the threshold before all the cells are dead, the population could recover and it would be just fine. And so we made a model that was in collaboration with Chris Ramin, a mathematician here, and we jointly advised the student Sia Vashriyazi, who came to one of these meetings. And don't worry about all these details, because I'm going to tell you in just a minute that this model's wrong. But we made a nice model that would explain kind of methanol changing, formaldehyde changing, growth, death, all those the kind of phenomenological behavior. And from this, we can actually simplify those 4 ODEs into 2 because of quasi-steady-state assumptions. And then if we look at a phase plane of formaldehyde concentration versus number of cells, we can see kind of a region that there's a null climb where d and dt equals 0. And so this is where death exactly equals growth. And if you're above that in formaldehyde, there's net death. And if you're below that, there's net growth. And so you could have populations start to die, cross that threshold, switch to net ability to grow, and that seemed to perfectly well explain what could be going on. And indeed, with this kind of model, we could take the data for both growth and viability, and it really, I think, captured the dynamics fairly well. However, the model was wrong, because the recovery was for the wrong model. We thought that this, again, that this change in being no growth to grow is because you're dropping the formaldehyde below some critical concentration. And that should happen right when this kind of inflection occurs. And instead, if you follow viable cells, they die exponentially, and then they grow exponentially, they increase in number, but formaldehyde has gone completely unchanged. Formaldehyde does eventually go down, but not until much, much later, until like maybe hour 70, but this inflection has occurred before hour 20. And so this model is also wrong, that there's not just, it's not just that they've made their world palatable before they've all gone dead. And so now we're left with trying to understand how can a consistent world that always has a high level of formaldehyde have things change from rapid death to permit typical growth rates.
Hey Chris, how do you measure formaldehyde in these experiments?
This is just with a NASH assay.
And is it good to within 20%?
Oh yeah, it's actually quite high.
So there's, okay, so you, all right.
Yeah, I mean, this is plotted on a log scale, and so it does compress things, but even still, there's no appreciable change until way out here. Can you see my cursor, by the way?
We can see your cursor fine. Yeah.
Great. Yeah, so it's really pretty flat. And so this was definitely a surprise, and it suggested, as nice as that model was, and as much fun as it was to talk about multiplies and quantum steady states and things like that, that the initial model of how this world was working was certainly not the description, because the explanatory variable is not changing. So what about
it's kind of funny how well you actually managed to fit the data with that model, the other data.
Yes, I think this is what's kind of scary, of course, about doing applied math. Right? All you're showing that this is a possible world that could recapitulate the data. Right? It's consistent with the data. It's one reason why I hate the model validation. Right? It's just not invalid. But yeah, so. But if you look at the data, it's basically one exponential down to one exponential up. And so you just need some model that would produce a sum of exponentials, and you could fit these data. But the question is whether you're fitting it the right reason.
Yeah.
And clearly, having it be due to external formaldehyde is not the right reason. So could this be something involving phenotypic heterogeneity? Are the cells that are dying actually different than the cells that are growing later? Or the cells themselves changing? And so perhaps this would be like antibiotic persistence. And this is where some cells in the population can change phenotypic state. So they can go from growing cells in green to non-growing cells in blue, and this can switch back and forth. And certain stresses like ampicillin will kill growing cells. You only have non-growing phenotypes around, but when you remove selection, they can rapidly change back to a growing state. And you haven't changed anything genetically, but phenotypically you've gone through this valley.
Hey Chris, does the lag time that you're observing depend upon the volume that you're subculturing into?
Yes, it does actually. So I mentioned before that if you go to kind of the real threshold, like say 5 millimolar, only some of the flasks will recover. And that depended upon what volume we did. If we did this in a 5 mil volume, it was extremely rare, if ever. And a 20 mil volume, it happened more frequently, and 10 was kind of in between. But I'll get to why I think that was. Yeah, but that's a great question. So we thought maybe it's something like this. And I think the key thing about persistence is this is a case that we know now that's kind of, it's bistable and has two discrete states. The majority of cells grow, all have reasonably similar growth rates, but they're sensitive to antibiotics. And a rare minority are just simply not growing at all. And they're resistant to many things such as ampicillin. Right? So it's two discrete states, and the ones that could survive aren't growing at all. And so if we had something like this, if there were two discrete states, so I've thrown some fake data on here. Say 1 in 1,000 were more tolerant, that they could somehow withstand formaldehyde up to say 5. But everybody else can only take it up till 2%. You have these kind of discrete thresholds. If you go to plate cells at different levels of formaldehyde, all the cells could take 0, could take 1, could take 2. And then only those rare kind of discrete state cells can take 3, 4, and 5. And then nobody could take 6 and above. Right? It looks something like this. However, our data look like this, that we get a continuum. 50% of the cells can survive 2 millimolar. And then each time you go up by a millimolar, you drop by something like an order of magnitude or more, such that only 1 in a million can survive 5%, 1 in 10,000, 5 millimolar rather. 1 in 10,000 survive 4 millimolar, etc. And so there definitely appears to be heterogeneity in the population, but it seems to be a continuous ramp up in terms of formaldehyde resistance, and not this kind of two discrete states that would be more like heterogeneity. So we began to ask whether this kind of behavior could explain the phenomena that we saw. And so what we're saying here is that if you look at tolerance, which is kind of encoded here with dark blue, with no formaldehyde, everybody can grow. Low levels of formaldehyde, you get cut off, and only above that can grow. Higher formaldehyde, only those can grow. And so we can model use of methanol, the use of formaldehyde as before, integrating over all those different tolerance types. But now the change in numbers is actually a partial differential equation, both with respect to time and with respect to tolerance. And so we have this continuous distribution of tolerance, and we have both an advection and a diffusion term for the tolerance. And so these tolerance levels can change, but there's a general trend to move them back towards lower levels. And this red part here is the death term, and what we're saying is if your index is above, if you're more tolerant than the formaldehyde level around, there's no death, and if your tolerance is below the formaldehyde around, then you die in a formaldehyde concentration dependent manner. And so it's relatively simple equations, just complicated a bit because of the PDE. So what does this look like? So again, this model can also capture these phenomena fairly well, where we see something like the death dynamics that we saw before, and we see something actually captures this kind of dynamic a little bit better even than before. And so what is being predicted that's going on? If we were to zoom in and just look at, say, the 4 millimolar curve, at time 0, what we're saying is there's this exponential distribution of phenotypic states, for most of the cells not being very tolerant to fewer cells being tolerant. However, most of the cells in this population get hammered quickly. They die at an exponential rate. And so it's only those rare cells that don't even leave very frequently in the beginning that by the time you get to this inflection point represent the cells that will end up growing. And then once they get growing up, all the cells have to be in this phenotypically tolerant state. And then long after, so this is 80 hours, if you let this sit and relax for 400 hours, the distribution will slide back. So that's the kind of dynamic that we're talking about. And this is what it would look like if you want to see kind of the fancy kind of heat mappy version of this, where this is the number of cells on the log scale, and encoded for kind of each of the tolerance levels. And so if there's no formaldehyde around, the distribution stays the same, but just everybody grows, and then nothing happens. If you go to high numbers, everybody just simply dies. So there's not, and notice the time is really short. So the distribution doesn't have time to really shift, and you get either slow growth or rapid, depending upon the concentration. But if you get this intermediate concentration, you go from this initial distribution to in between, you've got this bipodal distribution, and then later on, you've only got this higher resistance distribution going forward. So it looks pretty. Is this what's happening?
Hang on, back up one slide.
Yeah.
Why is there any change at all for 0 millimolar?
Oh, that's just exponential growth. It's just because all cells grew exponentially over the first 25 to 30 hours. And so the way right now that my students plotted this is absolute numbers rather than proportions. And so it's because the absolute numbers is going up for all of these, the color ranges change. Same thing over here, the distributions aren't changing, it's just the absolute number of everything is dropping. Including below, this would be the log of 0, so that's 1 cell. So he's plotting even, fractional cells. So it's obviously,
I see. So the breakpoint, in the 0 millimolar case, the whatever 25 hours is where they saturate, basically.
Exactly. This is when the growth curve is done, and so the distribution is the same. It's just there's a higher number of cells.
Okay, and then 3 millimolar is out by whatever that 80 or whatever that is. I can't see the numbers.
Yeah, exactly. By about 60 or 80 hours. Yep. Precisely.
Yeah. Cool.
So this looks nice, like you noted. I already showed you one model that looks nice. Why do we think this one actually might be doing a better job? We actually see this change in the distribution of tolerances. And so what's shown here, the gray is plating onto plates with no formaldehyde. This is plating onto 2, this is plating onto 4. The formaldehyde concentrations are down here in black. So again, you can see the formaldehyde doesn't change at all, and then eventually drops. But what you can see, what I've shown before, is simply in gray. You get this exponential drop and then recovery. But if you look here, this is that initial distribution I was talking about, where 1 in like, I don't know, 500 or 1,000 cells can resist 2, 1 in 10,000 cells can resist 4. That's your initial distribution. However, the 2s drop really fast, the 0s drop really fast. By the time you get to when we saw this kind of or around 40 hours, all of the cells are quote 4s. Every single cell there can resist 0, 2, or 4 millimiller throughout this whole time. And so it has indeed changed to this kind of distribution where everybody can take the high level. And there are some other interesting aspects that I won't go into now, but do suggest that these phenotypes involve some rapid switching. The moment cells even see methanol, you actually have a little bit of a drop in viability, and also very, very quickly, less than a doubling time, the number of cells resistant to 2 quickly jumps. So this is less than a generation, and so it suggests this tolerance can actually be induced. So there are some interesting, and same thing here, like the 4 millimolars, when you expose them to 2, this subpopulation kind of shoots up in number and then kind of settles back down. So there's definitely some more interesting dynamics that we don't yet have kind of active switching in our model. So how can we, so I think so far everything is really consistent with this idea, but what we'd really, really like to be able to prove is that the cells that make it and regrow are really a rare subsection of the population. And because you have a ton of cells, again, that lose the ability to make a colony and then simply regain the ability to make a colony, right? And so if so, if you put in some sort of genetic marker or otherwise, every single cell that was around earlier will also eventually be around later. And so what we've done, and this is data from Friday, is Jessica, who is Jessica Lee, postdoc in the lab, has done all of this experimental work. She has looked at using a dye that specifically stains membranes, and so if you stain them and then wash away the dye, all the cells will be stained, and the fluorescence doesn't go away unless the cells grow, and each time they grow, they dilute the dye by half. And so you could take a cell population, if you kill them, you put something like 20 millimolar formaldehyde, the cells don't pop, they're still there, but they don't lose their fluorescence at all. And so over a period of time, there's really no appreciable change in fluorescence. If you let them grow without stress, so if you treat them with no formaldehyde, they're just growing on 15 millimolar methanol, you get what I talk about, that as they grow, and the doubling time here is about 4 hours, as they grow, they begin to become less and less labeled because they're diluting it with new membrane. And so you can see this shift down, 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 and it slowly moves to kind of basal fluorescence levels without any of the dye around whatsoever. So if all cells grow and there was a lag, you'd see this red, and after a long period of time, this population would start to shift to the yellow, the green, the blue, etc. What did she see? At time 0, you have this distribution, but as you begin to have these later periods of time, you can see the green here, for example, you begin to see this hump. This part is just going to go down. These are the cells that they're not going to ever grow. And so this hump gets diluted down. This green both increases in number and continues to become further and further unlabeled. So that it becomes there, etc. with time. It's only a subpopulation of cells that were initially extremely rare that have already had many doublings by the time they ever became numerous enough to be detectable, and they take over the population. And so I feel like we now have a pretty clear picture, at least phenomenologically, with the idea that it's this rare population of cells that take this high-stress world, and there's this, whatever state, again, it's not a single discrete state, but there will be another population that can only take, and more of them could take 3 or could take 2, and even fewer that could take 5. So few that there may be one or none in any given flask, which is, and if you extrapolate our numbers, it comes right about to the number 1. In the experiments, we would see them sometimes recover and sometimes not. And so it seems to be that it's a rare population. They're in a phenotypically different state. However, that part makes it like persistent, but it's both different and able to grow. And if you look at this exponential rate, like it's a little bit slower while there's formaldehyde around, and then it speeds up a little bit. We've seen this repeatedly. The slope changes maybe by 5% or something like that. But but they otherwise seem like they essentially can take life with 4 millimolar nearly as if it wasn't there. And they're the ones that take over the population. And so what I think is really exciting about this, the experiments that both Janelle and Tomislav have done, and the samples that were generated to send to you guys, we're looking at this exact transition. And we were comparing what happens when you hit the cells with kanamycin to when you hit them with formaldehyde. And with kanamycin, we've got a couple things going on here. The optical densities are being plotted in blue or red. The circles are wild type, and the squares are the EFGA mutant, the one that lacks this formaldehyde sensor that stops you from growing if formaldehyde is there. And so when kanamycin is around, it doesn't matter really which genotype you are. The optical density continues to increase, even though you can see this is over, whatever, the first hour or two. There's a pretty rapid drop in, this isn't a super large scale. This is not plotted correctly. I'm pretty sure these labels are wrong because it looks like a log scale, and yet the labels are linear. I'd have to double check on that. I think this should be on a log scale. So there's rapid death and inability to ever form a colony, yet the cells are actually continuing to put on biomass, increase in optical density, and when we did our methionine assay, continue to incorporate methionine. What's really interesting is when you add formaldehyde, wild type cells, this is their OD, it stops almost immediately. And yet there's no drop in viability over this timeline. This is only the first few hours, and this is 5 millimolar formaldehyde. And so it's really quite different, even though they both inhibit translation. Kanamycin kills you, yet you continue to translate. With EFGA, you don't die, but translation does halt, unless you're a delta EFGA strain, in which translation continues and also viability goes on unchecked. So I think it's going to be a very interesting comparison between the two different scenarios. And then finally, these are the early time points. Given what we had seen from what I showed you before, we also continued the formaldehyde experiments with wild type long enough to watch not only do the cells kind of delay for a period of time and then eventually recover, we have some samples from these later time points from when they're actually starting to recover. So we can begin to learn what is it that's phenotypically different about these cells. We know if they lack EFGA genetically, they can grow in formaldehyde just fine. So are these cells just phenotypically have kind of abnormally low EFGA levels, or low ratio of EFGA to its binding partner peptide deformylase, or lower or higher levels of formaldehyde oxidation genes, etc. To begin to try to figure out what it is that makes these guys different from everybody else that does.
Hey, Chris?
Yeah.
The comparison with kanamycin is interesting. Have you looked at other things like, I don't know, redox agents or things like that where the cells could have an adaptive response, like hydrogen peroxide or something? I'm thinking about other things that would have a sort of potential for phenotypic variability, giving you a workaround.
We haven't. I think that's a great question. We did this experiment because there was a really nice paper from Heinemann's group in the Netherlands where they did really, really long lag times. So even E. coli sometimes has some really, really long lag times. For example, if you transfer onto succinate or transfer onto acetate, it can often take 40 or 80 or 100 hours. And they did this dye labeling technique and showed it's actually only a really rare subpopulation that grows out and not the majority of cells. And we also see only a subpopulation be able to grow if you switch cells from succinate to methanol. And so we had, for the longest time, people have studied that transition, including myself. I did work on that at my graduate school, where it takes about 8 or 10 hours to switch from succinate to methanol. And we've measured all sorts of things during that transition. And it looks like only about 3/4 of the cells, or sorry, 3/4 of the cells are dead. Only about 1/4 of the cells are alive and transitioning. And so when we're measuring whatever it is, transcript, proteome, metabolome, anything else, it's of course going to be some combination of these two very different phenotypes going on simultaneously. And now that we know that, I think from the proteomic and transcriptomic data, we can look for not only the causal, possible hypotheses for what the cause could be, but also genes that we can now make transcriptional and translational recorders to, so we can actually look for phenotypic heterogeneity in gene expression early on in this experience, and be able to kind of identify which are the cells that are going to make it. And were they different, for example, before the stress ever got there, than the cells that don't make it?
Things like these experiments just seem like they're crying out for a microfluidics strategy where you could look at individual cells. I mean, if you could survey 1,000 cells diluted out, then you'd have one that would just keep growing as you change the medium condition and maybe a few partially or something like that.
But cry out is a good way to state it. We indeed have that experiment going on this week. And so there's a collaborator here in physics, Andreas Fastekis, who does a whole bunch of single cell work. And he's doing exactly that. Even before we had the membrane guide data, we've been feeling more and more good about this kind of phenotypic heterogeneity model. But we really wanted one very solid direct measurement of what's going on. Either watching them directly, like you just said, getting this fluorescent dye to work out properly, or the third technique, which we're still going to go forward with, is to introduce a population of barcodes on a plasmid, so have like say 40,000 barcodes, and watch what proportion of barcodes actually survive bottlenecks, and how that maps out relative to our expected bottleneck size from these other techniques.
That's quite nice. So you get a direct measure of bottlenecking from sequencing.
Exactly.
What's the nature of the fluidic platform?
So in this case, so there's a couple possibilities. He does have microfluidic devices that would allow us to say even set up a gradient. So you'd have 2 channels, maybe one, both with methanol, but only one with formaldehyde in it, so you can watch kind of a gradient. But the simplest experiment we're going to do is simply put the cells between agar and the cover slip and just watch the microcolonies grow. And so he can watch and look at extension rates and distributions of cell sizes and things like that.
So it's a time-lapse video on microcolonies.
Yes.
And you can change the solution within that?
For that one, no. But for his microfluidic device, which we would try second, you can. Yeah. And it would be, it would be excellent to, with a microfluidic device, we could also play games in terms of giving like a small formaldehyde spike, and then even maybe take it away and then give them a bigger spike and ask kind of, can we induce this resistance? Questions like that.
Yeah. Does the small proportion, is this going to depend on the initial dense number of microbes or not?
That's a great question. It seems to depend on multiple things. Let me go back to this distribution. Its shape is very consistent, but it quantitatively is a little bit different. The whole curve shifts back and forth, whether you're in the exponential phase, whether it's methanol-grown cells or succinate-grown cells. And even if you played out at 25 versus 30 degrees, things like that. And so there are definitely factors that affect it, but the overall qualitative behavior is true across all conditions we've looked at so far.
A couple of questions.
Yeah.
So when you played for the CFU, do you actually play them on the formaldehyde version or just the viability? Just look at the LBD.
That's with formaldehyde in the plate.
So they are resistant to formaldehyde on plate as well.
They are resistant on the plate as well. Yeah. And we've done the same dash assay to confirm formaldehyde is still there on the plate at nearly 100% of the concentration we initially added.
And second question, if your initial inoculum is higher, would you get more of these cells? What happens if you have higher?
Yes, that's exactly what you get. It's because of that. It's something like 1 in 10,000 can survive for 4 millimolar. When we get to these 5 millimolar concentration, it's about a 1 in a million event, and our inoculum was approximately 1 million cells. And so at least so far, everything seems to be consistent with the idea that when we had a population either make it or not make it, it was literally, was there one cell at all, a population that could grow or not. With the barcodes, I think we're really going to nail this, because if that is true, it will be a single barcode that recovers.
But there's no evidence from deviation for your model that there's any herd effect, I don't know if that's the right word, some colony effect, biofilm effect, where the escapers are doing well because they're surrounded by individual microbes that protect them. Is there some kind of insulation?
Yeah. I would say the answer is no. We don't have any evidence. I think one of the really interesting things that we'll get from direct observation, and I think it's a great idea, which is certainly why we're excited to be doing this with Andreas. So in these situations, like whether it's where's 1 in 1,000 or 1 in 10,000 are making it, and we might have a chance of observing them. Those are the cells that survive and can go all the way to making a full-size colony. And the colonies actually end up coming up in pretty typical amount of time, despite the high formaldehyde around them. What I don't know is for the other 99.9% of cells, do they at least start to grow and just never make a colony? Do they extend their cell at all? Do they go through 1 or 2 divisions before they crap out? Or is it just nothing? They just don't do anything from time 0 compared to those that do. And so just looking at number, we have no idea about that. In October, I did present some data when I was out there about using scanner beds and looking at times of colony arising. And so we know that there certainly can be some interesting aspects about high levels of formaldehyde causing high variability in colony arising time. But those were, the setting was a little bit different there. But we just don't know, because we can't observe anything until it's some decent number of pixels, which are probably into the 4th, into the 5th cell.
So Chris, just to follow up on John's question, imagine a hypothetical scenario where some proportion, tiny proportion of the cells clump. And in general, they're individual cells, say, growing in solution, but 1 in 1,000 have a few cells around them, 1 in 10,000 have a few more, 1 in a million have a larger clump. So there's some exponentially distributed clump size kind of model. And the bigger the clump, the more the one in the middle is likely to survive the formaldehyde level. So
I think that's a really cool hypothesis.
I mean, I'm just thinking of ways to get sort of a herd immunity-like effect, where you still have a small proportion of survivors with phenotypic variation.
Great idea.
But that would not allow them to grow on CFU counts on formaldehyde selection. So that is
maybe if there was 1,000 cells on land. Yeah, if there was an exponential distribution of clump size or something like that, perhaps. So I love the idea because it's also testable, because we could, of course, then take the cells and filter them at some relatively, not too small of a filter size, a couple microns or something like that, and just ask whether it changes the distribution.
So that's a trivial test, right? Yeah.
I love it.
Wouldn't they grow out of it though?
Well, I guess the thing is that barcoding wouldn't distinguish that possibility from the phenotypic variability one.
But
I mean, that's
If the clump was siblings, if the clump was non-sibling, then it would look different.
But the single molecule, I mean, the single cell, video microscopy would distinguish that trivially, I guess.
Yeah.
But wouldn't they grow out of their little?
Well, they would, but they'd be protected initially, and then they grow and.
And what? But they're still in the concentration.
Yeah.
So if you
But maybe they're coated with other dead cells or something, right? They've shielded
As
dead
they
cells
grow.
as they grow. I don't know, you have to invoke something, I guess. So.
I mean, it does work with antibiotics and biofilms, so I think it's a really nice idea, and it's definitely worth looking at.
Have you ever plated cells without growing them in liquid culture with formaldehyde directly onto the plate with formaldehyde?
I think so.
Yes, I think I'm understanding your question. We've done this kind of experiment both at the beginning as well as while they're growing. Is that what you're asking?
Yes, so the idea is that if there are cells already existing which are resistant, they will grow directly, right?
And they do. And so that's this 1 in a million cell. And so,
being exposed before formaldehyde.
So you get 1 in a million colonies grows up.
yes. Yeah. So for example, at the beginning of the experiment, instead of counting 10 to the 7th, we might count 10 to the 4th or 10 to the 2 that can make it at 4 millimolar. By the time the experiment continues, we get the exact same count on 0, 2, and 4 millimolar. All the cells there are in the exact same phenotypic state, within reason.
So I have a different question here. So when you're looking at the genetic hypothesis, you evolved them in the formaldehyde and then went back and you saw the exact same phenomenon. So now that you have this information, do you think that's the phenotype being, so what your data now suggests that this phenotype is hard to turn on, but does putting them sort of back to the beginning suggest that it's easy to come out of that phenotype? Or
It does. I didn't show the data on this, but if you take cells at the end here, if you directly put them in liquid or put them in or directly on the plates, all of them make it at 4 millimolar. If you take this population and put it here in the beginning, it'll just grow. It'll just right up.
Yeah.
However, if you do that experiment after 2 hours, after 4 hours, after 6, etc., you begin to see the lag, quote lag, come back up, and this distribution begins to move back towards this distribution over the time scale of maybe 2 to 5 generations. It goes back to.
And so you feel that's the phenotype turning off, not just a very few rare undead cells outcompeting the tolerant ones.
Yeah, and the interesting thing is if you grow them, so if you grow them on succinate, and it turns off very quickly. If you grow them on methanol, they actually will keep a decent proportion of their tolerance. And so, like, what world they're in, like, there's definitely some kind of hysteresis to this, that, like, exponential cells kind of are definitely kind of force, apparently, kind of a phenotype that begins to get lost during stationary phase. And what exact growth, so it's the same number of doublings, but whether it was done on succinate versus methanol, and succinate doesn't produce formaldehyde, and methanol does internally, changes how quickly and how much that phenotypic distribution relaxes back to its initial state.
Thanks, Chris. This was really cool. Hey, you said you've been sending samples. Is that have they been getting? Is this with us for the proteomics or Barrick or?
Yes, I think all the samples. I'd have to check what Janelle all said initially to send to Jeff's lab, but there are cell pellets for I think basically every single time point of these kanamycin and these formaldehyde treatment, and we went ahead and did a fairly dense sampling. And we've been talking with them about doing maybe a little bit of like a single replicate and trying a few more time points to kind of see what the behavior looks like so that we can better choose where does things look the most interesting.
Okay.
We have both the pre-stress treatment, so these times before zeros before they ever walk formaldehyde, and then 0 represents with the addition of stress.
Gotcha, cool. Yeah, no, I was actually looking at the density of your sampling. It's really nice. And I was laughing on the previous screen, I guess the previous plot. Go one back. No, I guess the one we were just looking at in the previous discussion where you hit, yeah, those guys right there, yeah. Is the gap, like on the 4 millimolar, is the gap between 10 and 20, 30 and 40, and 50 and 65, is that when the single person doing it went to sleep?
Yes.
Yeah, okay. It looked like they were spaced out pretty well by about 24 hours.
I mean, this is only 5 hours between here and there, so you know, she did pretty well. But yes.
Oh, they did a great job, actually.
I mean, this was actually part of, I mean, when we first saw the very first data that I showed, this, the time points were kind of spread out, and there's enough noise, and we didn't have replicates. So this looked like it could be exponential growth, but it wasn't really clear. You know, like we definitely didn't have a very clear idea of what was going on with these time dynamics. And I would say one of the first things that I really feel like this is perhaps two populations is how cleanly the data looked like one set of data of death and another exponential set of growth. Like this is just going down perfectly exponential. This is going up perfectly exponential, and at exactly growth rate.
Yeah, no, it's remarkable how well they manage. It's very clean data at this point. So.
Yeah.
Awesome. Okay, other questions? Yeah, one more.
I'm just curious if you've ever tried starting a new selection or testing for higher tolerance in the population, like the 4 millimolar one. The idea that if it's a continuous distribution of resistance, then you might have even more resistant ones in the 4 millimolar at the end of that time period.
Yes. We haven't. It's a great question. We actually haven't gone back into this quality level. However, we have a number of strains. If we have some strains that have a number of mutations that can grow on 20 millimolar as a formaldehyde with no methanol around. That was the initial experiment that got us started down this whole road. And so the strange thing there is they grow at 20 millimolar, you can freeze them away, you can regrow them, say, on 6 liter methanol. If you try to put them straight into 20 millimolar, you get nothing. And so you have to kind of first get them going at 5, and then 10, and then they can make it at 20. So I think what you're suggesting is exactly right. That you need to kind of ratchet the population in terms of its resistance levels. Even those cells that are genetically able to take really high levels, they can't do it absolutely instantly. Yeah, so I think what you're saying is probably absolutely correct. If you look at these cells that are actively growing in the presence of about 4 millimolar formaldehyde, and if you tried them on 5, maybe even 6, you could push them out. But there might be some limit. I don't know that distribution without genetic changes is going to extend ad infinitum. But I agree with you. Great idea.
All right, thank you very much. And thanks for your patience while we ran around between rooms.
Not a problem at all.
All right. Take care. We'll get everything. You got everything?
I can handle it.
You got it. He's got it under control. Okay. All right, Chris, I'm going to say bye.
Thank you.
Good. All right. Take care.
See you later.
Yeah. How are you?
Is this like a place for funding that will? There's an extra 400 million for Alzheimer's. So we have an Alzheimer's grant in it. It's not